physics and molecular dynamics, we can enhance the sampling of the reaction coordinates that we're interested in. So now you can see in the first image there, in molecular training, molecular dynamics, now you will see the reaction is just remaining the same. It's taking a long time to close the barrier. So to enhance the barrier, we impose a potential to the system that discourages the system from revisiting already visited space. By so doing, the reaction can cross the barrier and start exploring the other while controlling the area. So we started employing all these techniques, cleaning natural compounds that were available currently here, and other projects structure that we used to manage the COVID in Tanzania. One of the compounds here, they are from one of the planted radiation barrier. So we screened against the acid resin, and we found very interesting results. We found that one of the molecules at the interface and protecting the virus from entering the cell. And you can see the figure here at the far right bottom corner. The ring in green is binding at the AIST receptor cells and inhibiting the viral cell entry. So it disrupts the interaction between the two cells. By then we did not have an experimental paper to collaborate with, but we trusted much our techniques and our approach we used, to the extent that we said, let us report what we have already found. 2 years later, experimental people, they validated what we found in experiment in computation. This year, they reported that Eductaion inhibited the spike protein of severe acute respiratory syndrome from coronavirus. This was a very good motivation for us that with techniques we used from scratch, we were able to develop techniques approach that could experimentally be validated. So this compound was traditionally used, so we took it to in silico and then we recommended to the community to continue to be used. We then moved to other plant extracts like neem tree, and it was also widely used. And our aim was to find something already in the market. So we studied this compound and we screened them as inhibitors of the cell virus entry for the SARS-CoV-2. We found some very promising compound from the neem, but these compounds are not developed that this would have at least an activity. Our in silico study suggested that this molecule here, sulfonylurea A, the structure you see, it's already has been studied for many years. And we found in silico it has activity toward inhibiting viral cell entry. At this point, we already established the collaborated with a research group and experiment who they have been working on live SARS-CoV-2 cells. So we buy this compound, we purchased, and said, please, can you work on this? Our in silico technique, they are telling us that this could be a potential. It was very active, even though not content. And we extracted the parent molecule. And we found also that they are even more active than even a single candle. Despite that, sulfonylurea A is active even though not potent. For the in silico technologies and tools that the pipeline we developed, it is a very good promise and progress that can help us to improve and design and develop other medicines for the future. And we tried to explore some field reaction coordinate, employing free energy calculations. We discovered that some other free energy and reaction coordinate are very powerful compared to the other. If you look at this one figure, B and D, you will see that there is a lot of fluctuations towards the end, for the first and the last. But in the middle, they don't show anything. So we learned many things. And then we moved to other targets to see this compound, could it be able to inhibit the viral replication and the polymerization process. And therefore we targeted the MPRO target of the virus. It was interesting to note that the extract itself were even much active. We did not have much sample to test it for the envelope or the ceremonial day, but we tested for the viral implication, and we found that comparing to a best will was even potent again, but not compared to the lens rate, was more active. The toxicity was moderately quick, so it was not much toxic. So we found that this is a good technique, this is a very good approach. So we found that it's very important as the intermediate interaction. So what next now? We are now working in a project called the Oratum Research. We are now deploying this project technique to find strong antimalarial compound. There is an interesting story then. I said, what is the user of this? We need to take some compound to the community if we have some promising approach. I then started focusing, working on prostate disorders. Here there has been, it's becoming a very big problem. Many people have been diagnosed with enlarged prostate, the BPH. So I utilized this technique and founded a compound that was effective. I formulated it and tested it also. It was effective. I decided to become it for clinical application. It is now in the first clinical trial, it is doing very well. So we are taking the compound from initial experiment to the community application. As I mentioned before, the other area of interest we are trying to develop, but we are at the very beginning. Having said so, I would like to say, asanteni sana, grazie mille, karibuni Roswell.
Thank you very much, Daniel, for taking us onto this journey together with you. Are there any questions? Otherwise, I may start with two. So,
yes.
I would like to know about the current status of the compounds you were talking about, luteolin and salvinorin A. So, because luteolin in this publication has been confirmed, basically, as you said. So, what is the current clinical status? Are there trials, and is it reaching the market?
For salvinorin A, because now we see that we needed to optimize it more. So we are now in the second stage of optimizing it so that we can maybe improve much their activity. For luteolin, we haven't worked much, but there are several other groups that they are working on it. So these are some. There is another that I didn't mention for the sake of time, but it has been widely used in Italy during the COVID. So after we published the paper, and people they tested it, and it worked perfect.
So you don't see a problem with off-label use because the clinical spectrum has already been confirmed. I see. Any further questions? I have a very short one because you mentioned the application of stereology to molecular dynamics, and this is something. I mean, stereology, as we all know, is a very universal tool. So basically, it's a theory that can be applied to everything in the universe. As far as I know, it hasn't been applied to single molecules. At least I don't know of any application to single molecules. Could you please elaborate a little bit on how you envision the application of stereology at the single molecule level?
Perfect. So, the idea in stereology is like to get the visualization, the imaging, so you can understand more things in 3 dimensions. Molecular dynamics is a powerful tool that can help to understand and visualize it could be a molecular material in 3 dimensions in a real time. If we are trying to understand how the molecule is working toward a certain protein, we can then try to understand over time, visualizing the behavior of the complex system. How it is interacting, not only in 1 dimension, but in 3 dimensions, trying to see how it is interacting and to predict if it could be effective or not effective. With this understanding and vision, I call ND as a theoretical tool.
I understand. So basically you transfer stereology from 3D to 4D at the time.
Yes, with time.
Okay.
Yes, with time to 4D.
Any further questions? This is not the case, then thank you very much again for your presentation and for the discussion.
Thanks.
Did I miss one?
Yeah.
Oh, please, sorry.
Okay, thank you. I saw some equations there in your slides. Dynamic equation. Yes, that one. Did you just apply it or you derived it to reach there, or you just applied that equation on your reference?
Okay. So the first one is the Newton equation of motion. The force equal to mass and acceleration. The force is equal to the negative potential. The second term after the mass, that is acceleration. The second relative position with respect to time. So it's the question that we start with, and that gives the dynamics. But now, in order for the system to start moving, we needed to have the potential of the system, which is described by this term. Now, like if you have water, you have oxygen and the hydrogen, they are bonded. So the interaction between the bonded terms, non-bonded terms, you need to describe. So you tell the system, now my system is comprised of X, Y, Z bonded terms and the X, Y, Z non-bonded terms. And then you can start computing the force. So you get the potential, the system starts evolving with time. So we need to have an integrator that will start solving this equation. Yeah.
Okay. Thank you very much again for your presentation, and we move on to the next talk.
Yes.
Which actually is mine.
Welcome again. For Stereology, I would like to invite Dr. Matthias and say yours.
Apple to PC transition. So thank you very much for inviting me, especially to Suleiman. It is my first time in Tanzania. I already enjoyed what I've seen so far. It's a great pleasure to be here. As you can see from the title, the organ that I'm interested in is the lung, and you can also see from the title, by training I am an anatomist and an electron microscopist working in Berlin at the Charité. In the background, you see one of the buildings of the University Hospital, and here you see the anatomy building in which I work. by stereology is extremely worthwhile because the functional capacity of the lung is directly reflected in its structure. And this structure has to be measured to be understood how well the lung performs. If you look at this, it's fixed diffusion law that tells you that for efficient gas exchange you need a surface area that should be as large as possible and a diffusion barrier distance that should be as small as possible. And when you look at the numbers for the human lung, you see that we have an alveolar surface area distributed over millions of alveoli that is in the range of a tennis court. But at the same time, we have a diffusion barrier, a blood-air barrier, that on average has a thickness 50 times thinner than a sheet of airmail paper. But how do we know that the inner surface of the lung is the size of a tennis court? Because, of course, as you know, we cannot measure it directly. We have to measure it indirectly, and this is what stereology is all about. So in a thought experiment, you could think of sticking needles into the lung. And when you stick a needle into the lung, you can count the number of times this needle intersects the alveolar surface. And this number of intersections is proportional to the surface per volume of the lung. And therefore you can measure the inner surface of the lung by this principle of needles, which of course in real life is test lines. And the person who first thought about measuring lung structure like this is shown here on the left-hand side. This is an image of 1963 showing Ewald Weibel. And in 1963 he published a book that was basically the foundation of lung stereology. At that time it was called morphometry of the human lung because the term stereology was not there, it was not even coined. So he called it morphometry of the human lung, measurement of form. The reason why he did this is Ewald Weibel, originally from Switzerland, spent a couple of years in New York with two Nobel Prize winners, André Cournot and Dickinson Richards, and they gave him the task to do anything on the structure of the lung that is of interest to physiology. And as we all know, of interest to physiology is numbers. The structure has to come with numbers, and that was what Ewald Weibel set up as a discipline, if you want. Now we call this discipline stereology, and we can define it as a set of methods to obtain these morphometric or quantitative structural data in microscopy. And we all know it has a solid foundation in mathematics, in particular in stochastic geometry. And by this, stereology solves two problems. The reduction in size, because we cannot measure the whole human lung, so we have a sampling problem, and also it solves the problem of the reduction in dimension, because in microscopy we basically have two-dimensional sectional images of, in real life, three-dimensional structures. So we have sampling tools, a cascade sampling tool giving each part of the lung an equal opportunity for being selected for the analysis, and we have measurement tools that account for the fact that we lose one dimension when looking at microscopic sections. So we use parameters on the one hand and test systems or geometric probes on the other hand, and the dimension of the parameter we are interested in plus the dimension of the test system that we use to estimate this parameter is equal 3, as we know. So in other words, volumes are felt by test points, surface areas are felt by the test lines, like the needles I've shown you, length is felt by test planes, and number can only be felt by volumes which are called dissectors. And the person who invented the dissector is shown here, Hans-Johann Gundersen, and he did this in 1984, and this was the cornerstone, I would say, of the next level of stereology, which is now called design-based stereology, because we now have a set of methods, a toolbox if you want, of unbiased methods, and therefore it's called unbiased by design or design-based stereology, because we don't make any assumptions anymore on the structure of interest, not on its shape, not on its size, not on its spatial orientation or distribution. So now we have these design-based stereological tools to characterize the lung further. If we again look at our parameters of interest and their dimension, volume, surface area, length and number, the lung is an organ where you need all of these parameters. It's not only about number, we need everything. We need the volumes, for example, of the septal tissue, of the alveolar region, or we need the surface area of epithelium and endothelium, the length of the nerve fibers along the airways,
And number, for example, is the number of alveoli or the number of a specific cell type within the alveoli. And derived from this, of course, we can also have other parameters like particle size, which is nothing else than total volume divided by total number, or barrier thickness, as I have shown in fixed diffusion law, which is nothing else than volume divided by surface area. So all the tools are there. The one tool that came latest was the estimation of alveolar number. I told you that the dissector was invented by Hans-Joachim Gundersen in 1984, but it took 20 years until we found the solution how to count alveoli in dissectors. And of course, the solution came from Hans-Joachim Gundersen, because he realized the problem is alveoli are not isolated particles that are countable in the dissector in the ordinary way, because they are all connected and they have openings. But he made this a virtue, saying that, okay, let's count the opening rings. Disregard the rest. So the number of alveolar openings is the number of alveoli. And this is reflected in stereology by a term called connectivity. And the estimator is called the Euler number, according to the Swiss mathematician Leonhard Euler. So the Euler number of openings in the network of alveoli is equal to the number of alveoli in the human lung. 20 years of work. So now we have the tools to characterize the human lung. So we know how many alveoli are there, 480 million. We know their mean size. We can then go to higher magnification light microscopy based on semi-thin sections and estimate, for example, the number of type 2 alveolar epithelial cells and their mean size to understand hyperplasia and hypertrophy. And we can go to the electron microscopic level and even apply the dissector at the EM level when we want to look inside those type 2 cells and see their secretory organelles in which surfactant is stored. They are called lamellar bodies. And we can estimate the number of lamellar bodies and their mean size and the total intracellular surfactant pool in the human lung. You can even take it one step further and look at molecules. When you want to look at molecules in microscopy, at high resolution you use immunoelectron microscopy, and there you use immunogold labels, for example, for a specific protein within the surfactant system called surfactant protein A. And then you can use what Terry Mayhew has called the relative labeling index. And with this relative labeling index you can check whether a labeling is preferential or not. And one step we took was we tried to officialize stereology in the field of lung research. And for that we basically developed guidelines on behalf of the American Thoracic Society and the European Respiratory Society. So now, as you could say, stereological principles are now officially released by the ATS and the ERS as the method of choice to quantitate lung structure. And based on these official recommendations, we in the last years tried to illustrate how they can be applied to animal models of human lung disease. Different animal models, acute lung injury, fibrosis, emphysema, pulmonary hypertension or asthma. What are the parameters of interest that you should quantitate by stereology, and what are the sampling designs that you should use for this? And I will illustrate this with one example, a case of where we look at the behavior of alveoli during lung injury and the eventual development of lung fibrosis. And for that we have to do one more step in lung mechanics. We have to understand that the lung is subject to retractive forces. And these retractive forces consist of two components. One is tissue elasticity and the other one is surface tension. Surface tensions occur at every air-liquid interface, so also at the air-liquid interface at the surface of our lung alveoli. And they follow Laplace's law, showing that the pressure that you need to keep an alveolus open is proportional to surface tension and inversely proportional to the radius of the alveolus, meaning that the system is inherently unstable, because small alveoli tend to collapse. And we all know of prematurely born babies that lack surfactant, and they basically have alveolar collapse in their lungs, and they cannot breathe without medical treatment. So we need something in our lungs called surfactant, a surface active agent that counteracts surface tension. Lower surface tension in smaller alveoli more than in larger alveoli, so that we have an equally low pressure all over the lung to keep the alveoli open. That's the main function of surfactant. And it's synthesized, stored, secreted, and to a large extent recycled by this cell type, the type 2 alveolar epithelial cell. And in that sense, the type 2 alveolar epithelial cell has been termed the defender of the alveolus by Mary Williams and Bob Mason. And it's defending the alveolus by producing surfactant, but also by being the progenitor cell type. So the alveolar epithelium is always regenerating from proliferating type 2 cells, which can differentiate either in type 1 or in type 2 alveolar epithelial cells. And this is of particular relevance when it comes to injury of the alveolar epithelium, which is called acute lung injury, or ARDS, acute respiratory distress syndrome. It's the same thing that happens during COVID-19 infections of the lung. The histopathological correlate is called diffuse alveolar damage, and it can be divided into two phases. The first phase is an early, acute, exudative phase, characterized by injury to the epithelial cells, to formation of edema in the alveolar spaces and alterations of the surfactant system. And the second phase, it's the chronic and proliferative phase, is characterized by proliferation of the type 2 cells, trying to regenerate the epithelium, a phenomenon called cuboidal metaplasia, and therefore, of course, a thickening of the alveolar septum, then, of course, making problems with gas exchange. And this will, in the end, either lead to complete restoration in those who survive the injury, and in other cases, to the development of lung fibrosis in those who survive the injury. So, in order to study the sequence, what happens from the transition of the acute lung injury into fibrosis, we had to go to animal models, and we used the bleomycin rat model, in which we studied actually this phenomenon. And the reason is because then we understand what is the result of injury to the alveolar epithelium. We know it's surfactant dysfunction, which will lead to an increase in surface tension and alveolar collapse, and we also know that if we lose alveolar epithelial cells, the basal lamina will denude. And then, if we have alveolar collapse, we expect that we lose over the other, and you lose the alveolar lumen with the airspace in between. We also see that the collapsed alveoli contain edema fluid in their collapsed lumen by electron microscopy. And then when we do lung function measurements, the important parameter is called elastance, which is a measure of stiffness of the lung. And the elastance is increased in the case of acute lung injury. But the funny thing is that when you increase the pressure in the lung, then you can decrease the stiffness of the elastance. That means you have collapsed alveoli, but if you increase the pressure in the lung, you can still open these alveoli and make them available for gas exchange. At the level of alveolar type 2 cells, we see hypertrophy of these cells, we see changes and increase in the intracellular surfactant pool, so there is an increase in the lamellar body volume, and we see that the intraalveolar surfactant pool is decreased, so it goes down, with a decrease of the surface active subtypes, and this correlates also with a number of open alveoli. This collapse is then sealed by alveolar epithelium. I will show this in another micrograph on another slide. And then when we do the lung function measurement here, we see that the stiffness or elastance is even further increased in the chronic phase. But if you increase the pressure now, you cannot decrease this stiffness, meaning that the alveolar collapse has increased, but you cannot reverse the alveolar collapse anymore. It has become irreversible. That's the major problem. And here we see the stereological data of these lungs. So the stereological data show that the part of the parenchyma, the volume fraction of the parenchyma, that is no more. We see that the volume of alveolar air spaces, of course, is then also decreased, so we have less volume available for gas exchange. We see that the alveolar number is decreased, meaning that the collapse leads to a loss of functioning alveoli. And this correlates also the surface area is decreased, so we have less surface area available for gas exchange, while septal wall thickness is increased, which shows again to fixed diffusion law that we have a loss of functional capacity in these lungs. So is this only relevant in animal models? Well, what we did is we looked at human samples from patients with idiopathic lung fibrosis at the end stage of you understand from an animal model where we applied stereology that alveolar collapse and collapse iterations are important mechanisms here in the development of fibrosis. We have an injury of the alveolar epithelial cells with surfactant alterations that first lead to a reversible collapse of alveoli. They can still be opened, but then the collapse becomes irreversible, shown in higher magnification here with the red arrows. This is the tracing of the basal lamina. And here you see another infolding. Here you see the basal lamina infolded and then back to the surface again. So this was an alveolus that is lost. And you also see that proliferating alveolar epithelial cells seal this collapsed alveolus. So the outlook at the end of my talk is there is new developments in electron microscopy that I think could be of importance to stereology.
site of a particular compound of your interest. Why Drosophila? People may be wondering, studying Parkinson's disease from an insect. Drosophila has been used more than 10 to 20 years now as a very tractable model for studying the human diseases. And this is because after the genome has been sequenced, it has become realized that in Drosophila you have at least 75% of human disease related from someone from Germany or from Spain, anywhere, and you get the strain you want. And it's easy to create the transgenic model as well. Looking at this picture, you'll see this is the brain of the Drosophila. So normally there's roughly 120 dopaminergic neurons in Drosophila brain, and they are located in 6 clusters. These 2 clusters can be seen from the dorsal view, but more on the ventral view, where you see these other clusters. So they are located more on this protocerebral region of the brain of Drosophila. So first, as I said, we had to evaluate the toxicity by allow us to know how these animals are able to climb a vertical tube. We use the recommended concentration of our natural products. And we use the concentration which were below the lethal concentration in our compounds, and this is the PD fly fed with L-DOPA. So in general, all the compounds were able to rescue the locomotor disability that was induced by rotenone in our studied flies. Later we thought of measuring the level of oxidative stress in the tissue of these flies. Then we measure by using the marker of reduce the oxidative stress that was induced by rotenone in the Drosophila. To further, we also use the molecular technique. Here we measure the messenger RNA in the brain tissue to target the 2 genes, this antioxidant defensive mechanism in our brain and in our body, a sodium catalase, and when the adenosine receptor 2 and MAO-B. And in addition, molecular dynamic simulation ascertained tosantin A as a potential inhibitor of adenosine receptor A through the interaction of these protein residues. So here are just an overview of the results that were found from the docking and molecular simulation. So these results were published. The first results were published in Journal of Neurotoxicology in 2020, and the second one, it was published last year in November in the same journal, in which I was a corresponding author. So to conclude, we can say that our findings suggest that studied flavonoids, quinone and cinnamon derivative have neuroprotective potential via reducing the oxidative stress burden arising from rotenone-induced oxidative stress, but also have improving locomotor ability in the tested animal after challenged by a toxin, but also the plant-derived natural product, in particular tosantin, at lower dose of adenosine receptor 2 and an antioxidative mediator in the development of anti-Parkinson's disease agent. Again, the studied pure compounds from plants have potential to protect brain cells from toxin molecule and hence reduce the neurodegenerative disorders. Thus, further studies to formulate the anti-neurodegenerative drugs from these compounds and the like are recommended. What are the challenges we are facing is that the natural products are isolated in very small quantities, so they limit their characterization, but also during bioassay can have a limitation. But again, the funding is a challenge in most cases of research. Some equipment, especially the powerful microscope, is also a kind of a challenge. But we also face a lack of direct industrial linkage. For example, now we have identified these compounds, how to go further in the next phase to be applicable in the society. So I'd like to finish by thanking my team members and also these two, Nelson.
Are there any questions or contribution?
One question that of course is obvious. I mean, you did behavioral studies, biochemical and molecular studies. Have you ever considered to apply stereology to look at the brains of Drosophila? Because I guess it's a very well characterized brain anatomy in general, so you could assess also the structural effects on the brain by using stereology.
Yeah, thank you, Matthias, for the very nice question. That's why in one of, the challenge I was facing, or we faced, is this on the structural parts. We wanted to do the immunohistochemistry, but we lacked this confocal microscopy for this to have really to say that these cells may be in the control and in the treated. So that was one of the things, and even the reviewer asked this question. So we are still working on it, but yesterday I was discussing with Tony, and he said I can, maybe not waiting for confocal, I can try other means to be able to work on this. So I have taken that as a good advice and recommendation. Thank you.
So, very nice.
Okay, yeah, that is a very good question as well. In one group, we carried simultaneously. Yeah, in one group we just give them rotten.
I wanted to see what is the change of the brain, especially some part of the brain, especially some part of the part to be evaluated by. I think you do not use the, the internet cytochemistry is fine. You have to do it. But some of the simple, like electricity, let's say, you can count the number of the cell division. You know the brain of this animal, and then you can.
Thank you very much.
Any question? No. Okay, thank you very much.
I would like to invite Dr. John Mbweni from Arusha, Tanzania, to make her presentation entitled Examining the Regulation of Neurotrophic Factors Expression in Humans.
Can you hear me? For some reason. Can you hear me, please? Okay. Thank you so much. And I would like to say thank you. I know everyone has scheduled, but you decided to do your schedule. And your recommendation, Professor Kaplan, about stereology, I think I'm thinking now to ask if we can plan to have a workshop so that we can learn about how we use stereology in our studies, because I have a workshop sometime so that we can know how to use stereology. So my story again is not about stereology, it's just about glial cell derived neurotrophic factor production in involuntary and involuntary muscles. So,
sorry, Dr. I can answer your questions.
Yes, please.
Part small system, you can go forward, and then after that if you produce some paper, and then your boss, your rector, the chief of the department, lead of the department, well, support to you. I think we can arrange such a workshop in any place. Thank you.
Thank you so much. So, my talk is about regulation of gliocell-derived neurotrophic factor production in voluntary and involuntary. So, what are neurotrophic factors? Actually, these are small molecules that support the growth, survival, and the differentiation of both developing and mature neurons. The neurotrophic DNF, I think we have heard about these neurotrophic factors because they are classic, and NT3 and NT4, and another group derived because the name itself says the origin and the function. So gliocell derived, this means cells of rats, and the function is like the function of this neurotrophic factor, but one is from R&D systems, which says GDNF is a small protein that potentially promotes the survival of subpopulation of neurons in both the central nervous system and the peripheral nervous system. Another definition, this is just a cartoon. Actually, I tried to put my idea on a cartoon. So we believe that when it is released from the target tissue right here, the skeletal muscle, and it will go to, it will bind to the receptors, and will go up to the soma of the motor neuron, and there will affect about maintenance of the motor neurons or do repair or survival. And these are just showing the neurotrophic factors family, and everyone has its own receptor here, and they do crosstalk, but all of this is just to lead them to affect what we say here, survival and repair and maintenance. So, but why should we change a motor neuron when you put just a control molecule and you have GDNF here, but the motor neurons will tend to grow towards GDNF. So this means GDNF can be chemoattractant to the motor neurons. As a therapeutic candidate, how can it be affected as in all other tissues? So that's what I wanted to know. Different in different tissues, so that when it is used as a therapeutic candidate, we know at least in the other tissues what are cell lines. So the first thing was to know if these cells are expressing GDNF, and if they do so, how it is similar or different in terms of at least the concentration or corporization and all of that. And then I wanted to activate these tissues and see how much they do in expressing GDNF. And then I activated by new sure, we say differentiated cells, my tube, so you can see they make the skeletal muscle. And here are cardiac muscle cells from atrium, and you can see these are just non-differentiated, they are still single cells. And then the differentiated ones are here. Stimulate the skeletal muscle, even heart muscle, they can stay in the culture for a long, long time. For that, I don't know why, but they want to be stimulated with the stimulation. So I could collect the sample in this timeline. As I said, first of all, we wanted to know if we have this GDNF in these cells. So these are skeletal muscle. So we can see the red is myosin, green is GDNF, and this is overlay. So when we see the overlay here, this means that we have the colocalization with myosin and GDNF. But for the nucleus and the skeletal muscle, again, we found the differences, the little differences, because the things in the cells, they secrete almost like the same. But for releasing GDNF in the media, we found that the cardiac cells release more GDNF in the media, and the skeletal muscle just release just a little bit. And even so, this is just like the same, showing that skeletal muscle keep more GDNF inside and release just a little bit, and here release more. But here you can say maybe because skeletal muscle is contracting by demand, that's why they need just little GDNF. But for this one, who is contracting all the time, maybe GDNF is needed. Here, as I said, I wanted to treat the cells with Eductaion and add acetylcholine. And when I added acetylcholine, I found that when you add acetylcholine, GDNF goes down. And when you do that, of course it goes up like you see in the control. And I wanted to make sure that if this is true, so that I block acetylcholine receptors with the alpha-1 capsid, and now GDNF went back in the control. So this means maybe it is true acetylcholine is affecting GDNF, and this one is translated by thinking this cartoon I showed you, because when the muscle cells think that they have let's remind you what the hypothesis is. And for cardiac cells, I treat them with acetylcholine and the natural treatment. So you can see here when you have norepinephrine, you do have norepinephrine, the GDNF goes up, and with the acetylcholine, GDNF goes down. And this may be true because the parasympathetic system always they make the heart to slow down, and norepinephrine make the heart to beat more. So this you can translate this by saying GDNF production levels in cardiac cells may be corresponding to the intensity of activity. Stimulation, this is just quite different because you can see in skeletal muscle, GDNF goes up, and in cardiac cell when we stimulate, GDNF went down. And this was just for one house stimulation, but when you increase the frequency, it changed even though I'm not sure here because of the time. So, but that's what I was looking for, differences. So, the take-home message here is when we are trying to make like a therapeutic agent of the other chemicals, we need to make sure that we taste and we see, because we can just focus on one thing, appear to depend on duration and intensity of activation exposure. Acetylcholine effect in the skeletal muscle may respond as though the innervation is restored, decreases of GDNF expression. So, understanding the similarities and differences in the regulation of GDNF production in different tissues may provide a better understanding of how GDNF production may be modulated.
You had a higher rate of secreted DGNF in the culture medium. So, what is your idea? How does the GDNF get from the nucleus of the cardiomyocytes into the culture medium?
Can you you open and can you talk with the microphone?
So
Dr. John.
The question is. So the question was, you found the GDNF in the nucleus of the heart muscle cells, the cardiomyocytes. And you also found high levels of GDNF secreted in the medium in these tissue cultures. So how does the protein...
I have one comment. Thank you very much for nice presentation, Dr. John. Yeah, as Prof. Matthias suggested, if you are using the culture, you have to do some of the stereological analysis, especially it's very, very easy. You have to take the picture and then using some of the picture for validation. The other way, stereology technique books, I hope it will be published in next year. And then tissue culture is something new.
Thank you very much for a very nice presentation. It's not questions, maybe a comment. Professor Matthias is asking, have you used any chemical for disrupting the cells wall?
Oh no, it was just a normal production.
You haven't used any chemical for that, only vacuum? No. Okay, thank you.
Okay, Chairman, can you conclude to mark the end of this session? Chairman, Mr. Chairman, you conclude? So we are done with the first session. Thank you very much, Matthias, with your team, and also the presenters for the first session. And I want to make a few. Okay, so please, as you sit there, also moderate time. Because I think there is something we want to do today which is not in the program. Yeah, it's really after the break. After tea break, when we come back here, please, I want to read the following. After tea break, 10 minutes from now, please just proceed to the high.
Type 2 diabetes is a chronic metabolic disease that causes long-term complications in carbohydrate, protein, and lipid metabolism in many vital organs as a result of insulin resistance. While this disease was until recently defined as a condition seen only in adults, it is now frequently seen in children. This indicates that the disease will become a more common health problem in the future. Considering the long duration of treatment and the numerous secondary complications, type 2 diabetes poses a burden on the healthcare system worldwide in terms of both financial and manpower. At the same time, the fact that the diabetes is seen from the patient's perspective as having a lifelong and worsening prognosis causes patients to be negatively affected physiologically. All these reasons, it is extremely important to conduct studies on natural anti-diabetic products that will alleviate the whole diabetes. In diabetes, as a result of impaired insulin secreting among them is the increased free radicals. Studies show that diabetic patients are sensitive to oxidative stress. It is known that increased free radical tissue as a result of high blood glucose levels exacerbate lipid preservation. It has also been reported that intracellular antioxidant enzyme activities decrease as a result of diabetes. The goal of the therapeutic studies is to minimize and delay complications caused by diabetes and increase the oxidative damage using to strengthen reactions. Experimental studies shown that plants containing luteolin and this glycoside may be protective against cardiovascular disease by reducing the blood pressure and cholesterol. In diabetes, by beta-amyloid production, inflammation, and oxidative stress. In this study, it was aimed to highlight the deleterious effects of diabetes on the cerebellum to investigate the possible effects of luteolin. But today only the pathological part of this study, because this project is ongoing. Material and methods, when we look at the material methods, the experimental part of our study was conducted at Bolu Abant İzzet Baysal University Experimental NM-AIST Application and Research Center. Grants included in the study were obtained to Abant İzzet Baysal University, and we chose the Vistar albino rats. The animals were kept fed formed in the study. The first group is control. We gave physiological sign to the control group. During the experimental process, it is the 28 days. And for the diabetes groups, the strat buffer, and we gave it 50 milligram per kilogram. And strat buffer group, also we give those diabetes and luteolin. We gave the luteolin to the diabetic rats at a dose of 20 microgram per kilogram, and luteolin we solved in the dimethyl sulfoxide, and so we gave the luteolin 20 microgram per kilogram. Because of the salt, luteolin, the dimethyl sulfoxide, we want to ignore the effects of the DMS oil, and so we add these groups, dimethyl sulfoxide groups, and throughout the experimental procedures, it's 28 days. To create an experimental diabetes model, a single dose of 50 milligram per kilogram streptozotocin was soaked in strat buffer, injected to experimental animals, and feed intake was allowed after streptozotocin injection. At the end of 72 hours after streptozotocin administration, fasting blood glucose volume were measured by dropping blood samples taken from the tail vein of rats that had fasted the night before onto a glucometer strip. After 3 repeated measurements were taken from each animal, animals with a fasting blood glucose level of and body weights and fasting blood glucose volumes of animals in all groups were measured at the beginning and end of the study. After the cerebral tissues of the animals were removed under 10 milligram per kilogram xylene and 90 milligram per kilogram ketamine, and they fixed in the 10% buffered neutral formalin solution, and the other procedure followed and embedded in the paraffin. And also we took the 4 micrometer fixed section from the paraffin blocks, and we took them the systematic random sampling and the histopathological analysis using a microtomic device. We take sections and we stain them, and after we do histopathological studies, analysis. Evaluation of diminished cerebellar tissue was performed using 5 different areas were selected, and these morphological parameters were scored semi-quantitatively between 0 to 3. If there was no change, the score was 0. If there was a change of less than 25%, the score was 1. If there was a change of between 25 and 75, the score was 2. If there was a change of more than 75%, the score was 3. In this context, quantitative results were obtained by comparing between groups. Each parameter was determined by the same each tissue sample belonged to which group and by random selection from tissue samples. It was the blind evaluation. Our statistical analysis there was no significant has neuroprotective effects on the degeneration of the cerebellum as a result of diabetes. We believe that the present study will provide new ideas and experimental clues for further studies on diabetic cerebellar image and treatment related to blood glucose levels. In addition, further studies conducted in different laboratories with different experimental elements and different methods are very important in finding out the possible effects of diabetes in more detail. Thank you for your attention.
Doing stereology only?
Yes, I will do also stereology, molecular analysis, and biochemical analysis. It's on Android. Thank you.
Okay, please.
Yeah, thank you for the next presentation. But what was your positive control setup?
Positive control, even when I do immunohistochemical analysis, I will have.
No.
Only control.
No, I'm asking the positive control for your doctor.
You are right. Yes. I understood too, because I guess I will. But when I look into literature, I guess because of we are doing injection and it triggers the stress. So I have to give saline. But when I look at, they say no differences. So I accept it like this and only this control.
Next presentation about stereological approach in amyloid and stereological imaging dataset of some urogenital system from Berlin, Zohar Altunkaya. Video? Yeah, video.
of
Hocam ses verler aslında.
can be easily determined by the Cavalieri principle, and also this estimating of the numbers of the polygons for all sides. We would like to preview this from the same study titled by Stereological and Histological Evaluation of Ovary and Referred Models of Female Renatally Exposed to Dicarbonate Sodium. In this image, we have histopathological images of the control and study groups, and also we have the volumetric data of the same groups, and both of the data was submitted as a result of the metrics. The test is another organ of the female system, and it can have another study about the placenta. In this study, the volumetric changes of the placenta, because for the different pregnancy complications were investigated. In this study, we have some volumetric data and histopathological images, and in the results we submitted both of them to the scientific journal. This is a major study, and it has the vital role in the male reproductive life. In the literature, there are many studies methods. As astrological methods, we can use again the principle and dissector, tubular diameter, and tubular volume, and the estimation of this cell numbers.
To avoid bias. After this, by using the geometric rules, we can estimate the volume, surface area, and other properties of the tissues. Stereological methods can be used in many areas of the reproductive biology studies. These methods can give us important information about the folliculogenesis, placental development, embryo morphology, testicular structure, or fertility assessment. As you know, various factors such as age, lifestyle, and environmental factors can influence the number of reproductive cells. The number is a very important factor in the human fertility. With stereological methods, it is possible to analyze the all cells of organs or the number of a particular cell type in a tissue. And also, the measurement of the volumes in a tissue is one of the most fundamental applications of stereology. We examined, such as ovaries, uterus, or testicles, are obtained from the experimental animals or humans. Then the fat and connective tissues are removed. The tissue is then immersed in a fixation solution and prevent bacterial activity. We generally use sections are stained to enhance contrast. Finally, sections are mounted on a glass slice for observation and analysis. Image analysis can be done by capturing images using a microscope and digital camera. However, images are calibrated to accuracy and consistency during the analysis. After this, spatial software is used to analyze the images and calculate the volume, surface area, and property of ovary, uterus, and uterine horn that we can calculate with statistical methods such as volume of the ovarian follicles, interstitial tissues, and germinal epithelium. In addition to this, we can estimate the number of different ovarian follicles. Estimation of the volume density and the total volume. In the image, you see the pluses on cross-total volume. In the image, you see the pluses on cross-section uterine horn, luminal volume, and volume density of the uterine glands. No matter what it is, you can find the counting frame is superimposed on the images. The tubules, which are either complete or partly inside the counting frame and are not touched by the exclusion lines, are counted. Estimation of the seminiferous tubule or V-size diameter. Diameter of the seminiferous tubus is estimated on the tubus sampled by the counting frame. The largest diameter to the orthogonal to the longest axis of the tubule and approximately touching the center of the tubule is considered as its diameter. Evaluation of the number. Two approaches, including the optical fractionator and volume-based ratio technique, can be used to estimate the number of the cells. Both methods rely on the dissector principle. The term dissector comes from the composition of the terms deep, pore, tube, and section. One section is called the reference section, while the other one is called the lookup section. There are two types of dissector method, physical and optical dissector. The physical dissector methods, this makes use of two parallel thin sections taken from the same tissue blocks. We need to use sections of the tissue. Consecutive serial sections are created by systematic random sampling. It provides computation for the pool. As mentioned in the previous presentations, a big size particle is given more profile than the small size. But Stereology does not affect this problem. Because of the basic idea of the dissector counting method is to count the top of the particle. In this reason, in the dissector method, the calculations are not affected by the size and orientations of the lookup section. You can follow it by the yellow arrows. We consider this profile as the top of the cells, and it must be counted as a dissector particle. Another important point that you should keep in mind here is that the quality of the section preparation should be high. If you cannot sufficiently visualize the structure within the tissue, you cannot make an accurate count. Optical dissector method. This method is actually a derivative from the physical dissector. It can be regarded as a three-dimensional or optical analog of the physical dissector method. In optical dissector application, there is no need to use two physically separate sections. Counting can be performed via optical focusing throughout the section in a relatively thick section. The main principle of this technique is count the particles directly in relatively thick sections using suitable lenses with the high numerical aperture. Therefore, first we need a thick section to apply an optical dissector. Second, an apparatus suitable for downward in the Z direction. The microcatheter attached to the stage of the microscope will measure the Z-axis movement. Additionally, as seen here, two guard zones are defined on the upper and lower surface of the sections in order to avoid physical defects that occur during sectioning. Counting is carried out in the middle part other than this distance, for example, within the safety distance, leaving a specific space on the lower and upper side of the dissector. Critical points applying to optical dissector. Thick section should be used. We generally use 20-25 micrometer for the optical dissector. When using a thick section for particle counting, it must be remembered that the entire thickness must be properly stained. If any part of the section is left unstained, the particles cannot be seen and counted. Guard height should be also left from the avoid section defects. And also, before counting begins, counting criteria must be determined. Usually, the nucleus or nucleolus is used as a counting criterion. Another and very efficient way of the estimating number is fractionator principle, which avoids the reference trap and is independent of tissue deformation. Is fraction-based method. This method is based on dividing its structure into parts and applying it to these.
In practice, the tissue is split two halves. The two halves are embedded in a block and sections are taken from the tissue block. Fractionation is done as follows. First sampling fraction is sampling, section sampling. The aim of the sampling step is obtained to 8 to 12 sections by systematic uniform random sampling of a known fraction of the testes. The second sampling fraction is the area sampling. It is the fraction...
Sorry, your time is finished, because...
Last slide. It is the fraction of the area of fields of weave in relation to the total area of the section. The third sampling fraction is height sampling. It is the fraction of the dissector height in relation to the thickness of the section. Finally, the total sampling fraction is calculated by multiplying all of them. In conclusion, strategical methods can be easily applied to reproductive organs. The obtained findings can be used in the diagnosis of infertile individuals or histopathological evaluation of the risk.
We have to follow. Next.
Yes. No coffee break?
No.
Yes.
Now we are moving to next presentation.
I come from Nevşehir Hacı Bektaş Veli Üniversity, Nevşehir, Turkey. I am happy to be here. I would like to thank you, Prof. Kaplan and other Congress members. And as you know, the central nervous system contains the brain and spinal cord, and the brain is responsible for responses, emotion, memory, communication, through processing, and the spinal cord, which is a vital aspect of the central nervous system, both found within the vertebral column. And the purpose of the spinal cord is to send motor commands from the brain to sensory organ, as well as to relay sensory information from the sensory organ to the brain. And why is it important to estimate the volume of central nervous system part? Researchers want to some analysis of the volume. Why is we want to estimate it? The brain volume is not only brain, all central nervous parts volume is an important data for several disorders in both children and adults. Evaluation of volume change of gray matter, white matter, cerebral, spinal fluid provides information about physiological process, presence, and severity of disease. So the measurement of brain and spinal cord volume can be counted as an objective parameter for neurodegenerative disease. At this point, researchers concerning several disorders such as autism, schizophrenia, hyperactivity, multiple sclerosis, Alzheimer's disease, and Parkinson's disease highlights the alternation in the nervous system parts volume. MRI is the most commonly used imaging method. MRI technique provides detailed visualization of the human brain in vivo and is widely used in this. In this technique, the proton Cavalieri method. Cavalieri method is based on the principle of the Italian mathematician Cavalieri. It is an impartial method of calculating the volume, regardless of the shape and size of the structure. In this method, the desired structure to calculate to make the surface area. The area estimate of each section facing the same side are done, and the result multiply the section thickness and volume of each area. This step is performed for all tissue fragments, like this. The volume of structure is achieved by collecting the characteristic of the structure to be calculated. When structure that show unclear or complex navigation need to be used with shorter distance. With smooth boundaries like this are marked and the advantage of OSARIX program is presence its integrated picture archiving and communication PACS system, allowing the automatic storage of the patient data. If the image cannot be taken from the system automatically, it's offered to change to upload it manually in DICOM format. OSARIX is a program special plane projection, convolution filter, variable slice thickness adjustment, volume rendering, minimum and maximum density projection, and surface rendering are also available in semi-real time, depending on the situation. The area intended to measure is framed in JPEG or DICOM format are transferred to ImageJet. After importing images into ImageJet, all analysis can be done easily and quickly in 3 steps. First one is the arranging the scale of the image, other one is the marking the area of the interest, and for performing the calculation. Whose boundaries are not fully understood, as well as the software toolbars. However, it's unlikely to be analyzed manually. In addition, point counting method is more useful in structure whose boundary cannot be clearly distinguished and which are difficult to distinguish. It's a fairly complex issue, and this assessment provides the data for many qualitative studies with the active systems inner atlases. The area of the brain surface is structured by superimposed to the obtained image of intention in the program is to extract the skull and scalp. After the removal of the brain tissue, imaged by magnified chains are removed. The density restored brain divided into basic tissue type by voxel-based tissue classification. Then, the program then applies the topographical correction to remove topological inconsistent in volume. The completes the segmentation process using processing and editing function. The BrainStream program, which is considered to very advanced in clinical study, is also used to determine the architecture of the brain. While this allows the extraction, the bone structure, the of vessels, and monitoring of aneurysm, the main artery, its colorated branches. In the image created through the image combined using the program options. The resultant image can be rotated, allowing the surgeon to better understand intraoperative aneurysmal anatomy. In addition, it is very effective and fast in obtaining volumetric result of the area. However, for using this program must be perfect. In conclusion, even minor change in brain morphology can be significantly affect in brain function. For this reason, imaging of the brain and analysis of the image play an important role in the diagnosis and treatment of many diseases. In volume analysis to be performed on MRI, methods
Rafael Mwezi? Rafael Mwezi, okay, around. Have you lowered your presentation here? Arife? She's here. Yeah, I'm just saying if the presentation is here. Arife, your presentation is here? Can you put your presentation here? No. Please, let this be here. Mohammed Musa Isa? Please, John, come here to the presentation. Yeah, please. Okay. Please let your presentations be here now. Those who are not for here. Murat. Murat Murat. And then Omar. Omar. Presentations be here. Omar. Is it here? Barker, presentation, absent. Amira, presentation for Amira. Amira, mind. Those who are presenting, please let. Again, those who are presenting, especially the first group, please let your presentation be before the coffee break. Maybe Asuna is here, Rafael is here, Rifa is here, and then Mahmoud is not here, is it? Mahmoud is not here. So we have all presentations here. If you think you are in this session, please come in.
Seni bilgisayara getir Hasan. Seni bilgisayara kapı getir.
Bilgisayar lazımsa vereyim. Getireyim mi?
Okay, I think everything is okay now. Learning. Okay, thank you very much.
Next presentation belongs to Dr. Nokunda. He will present the presentation as a video.
was that the brief background of the subject. Research has generated evidence that links low level of fluoride exposure and the benefits to human being and bones. However, if this level is elevated, then exposure will lead to tuberculosis and scoliosis. There is no agreement whether fluoride in the drinking water is toxic to the developing human brain. Researchers have carried out a research on this subject, and some are supportive while some are not supporting this phenomenon. Further research work have been undertaken towards understanding the water fluoride problem in the Eastern Africa region. And this backs to the year 1953 when Neville and Brass and also Williamson did some studies along the Eastern Africa Rift Valley on the undesirable effect of fluoride on human teeth. And since then, several research has taken place, and since the establishment of NM-AIST, team has been working much on the issue of fluoride, especially on the issue of defrogation. Despite that, there have been a number of research taking place on this subject. However, there are still knowledge gaps, uncertainties, and fragmented data about the entity and the impact of the fluoridation phenomenon. So there are several pathways for communities to get exposed to fluoride. The main source of fluoride is the rocks. Areas where there are these rocks that contains fluoride, when they interact with the water supply, then the water will pick up the fluoride, and these are available to the human and also livestock. WHO has set a limit in drinking water is 1.5 milligram per liter. Yes.
Sorry, please look at the TV screens.
The communities. The most important pathway is about drinking water. Another pathway is also food crops and animal products. So there have been a number of interventions that has taken place in Tanzania following the various research that has taken place. This includes the fluoridation of drinking water with household filters.
I think we can pass this one. It's not suitable, you know. Move the second one. We also release this video in our webpage. Announce the second one.
Our other speaker is the Rafael Merizeli. Topic is the pesticide use and brain health effect in communities living in areas with agricultural activity in north region of Tanzania.
If you want to use the free microphone, you can use this one. Do we need the free microphone?
It's okay.
That's okay.
So my topic on the effects of the organophosphate exposure with effects to acetylcholinesterase and sleep cover in mice model. I'm here with Sister Bembeli, Matunda, and so by introduction. Pesticide in Tanzania, global pesticide has been reported to cause serious adverse effects for human health. And the data show that around 200,000 cases are reported specifically on active poisoning cases. And all these deaths are happening on developing countries. And the WHO report on 2017. On average, 700 cases of death happened in India, reported in 2006. And in Tanzania, a report by Dr. K in 2016, it showed that only 15.96% of all pesticide acute cases are reported in Tanzania. So you can see pesticide is very problematic in our country. This is some of the pictures happened due to the misuse of pesticide, the occurrence of pesticide poisoning occurred by mishandling of the pesticide. And in 2016, the main pesticide has been reported to cause severe drugs of antidotes like atropine, 2-palm, and diazepam. These are controlled drugs which are very expensive to treat the pesticide exposure. So the study aimed to develop the rope antidote, which can handle by the alternative of using these controlled drugs which are available in hospitals. So, and the literature shows that the use of phosphate pesticide induces radical species which induce lipid oxidation, increase of antioxidant enzyme, induce neurotoxic actions, and the inhibition of the acetylcholinesterase. So the target here is to have the extract which can act as alternative to encounter the risk which is basically categorized from the cholic to acetic acid. So the phosphate block the pathway of the acetylcholinesterase enzyme eventually causing the adverse effect to human health. So the study was to find alternative how to replace the binding effects of the phosphate by using the extract from this plant is called the Acacia prolifica, in Swahili called mbunga, or by local tribe Masai called chlorite.
It's a very common plant here in Tanzania, especially in northern parts, and so many studies have been identified to have a lot of antioxidant activities, specifically flavonoids and a lot of polyphenols. So the target is the availability of these antioxidants to scavenge these lactose species so that to prevent the damage from exotic organisms. So the idea, as I said, that due to exposure and not farmers to afford the idea comes to extract the extract from the plant and to use as alternative to treat the exposure due to the use of organophosphate, specifically of chlorpyrifos, chlorpyrifos, and benzene. So the study has said to contribute on the development of antidotes for the treatment of organophosphate pesticide exposure. So the idea, we had the objectives to extract the good extract from the plant by using methanolic and to assess the reducing power, which is one of the assessments of the antioxidant property, and also to determine the acute toxicity, and lastly to do the assessment of the recovery of the antioxidant activity by using three pesticides, chlorpyrifos, chlorpyrifos, and Eductaion on mice model. So in the methodology, the extraction was conducted. I mean, the collection of the plant was taken from the monopoly. The first one was the plant extraction, and then we do purification before going to the objectives of assessing the radius, oral toxicity, and recovery. For this, the interest will be based on this for extraction process, as I said, and this for evaluation of reducing power of the plant to assess the antioxidant property. So the reducing power was tested by using these machines called UV spectrophotometer to assess the capacity of the plant. And for the model, we use albino mice to assess. So before that, we received the echo clearance from the KSRI, and also we conducted the oral toxicity of the plant at the different dose by using the different concentration, and also before introducing the plant, we conducted pilots to identify at what dose the mice can be exposed with the pesticides without being harmed. So we conduct the pilot and determine that at 3 milligrams or 2,200 ml of water, the mice can be exposed but not be harmed or cause the lead effect to them. And this is a device, it's called acid estimate kit. We quantify the level of cholinesterase. It's a rapid test. It's almost 5 microliters of blood were taken from the mice and analyzed in the device. And the setup was like this. 30 ml of chlorpyrifos in 200 ml of normal saline were diluted and exposed the mice. And the exposure was 28 days. The first group was the control group, and the second group was the pesticide plus extract. The third group was pesticide with ascorbic acid, and the last one was the pesticide with normal feeding. So this recovery was by normal feeding, and the third group, the pesticide which we use here is called chlorpyrifos. So we observed the effect from day 1 up to 14 days, and then it goes up to 28 days, where we found the effect. So from day 1 up to 14 days is just for poison. We poison them by chlorpyrifos, and then we look at the effect
After 21 days, we remove the exposure and we do the treatment. This for another for propylglycol. This is for propylglycol. The same what we substitute here is the pesticide that we use here is propylglycol compared to previous one where we use propylglycol. And this is the last one we use the Eductaion pesticide. The same setup. What we change here is just for the pesticide that we use, but the setup remains the same. So for the depression, from day 0 up to 14 days, the pesticide, the level of the pollinators for the rice were decreased, but slightly increase of the pollinators was extended up to 48 days for the Eductaion exposure. But the variation of the age from the sex and for the male and female were also 14 days after we introduced the treatment. Gradual increase of pollination response were observed. So this was done by crude experts, but we comment that for the further study, the isolation of active compound is very necessary, and also other plants within which are dogs that also encouraged to be assessed, and also further studies on serology to assess the tissue and other effects also are recommended for the further year. So we managed to publish this on the effect for this unit on accessibility, and also we thank the African Development Bank for funding and the quality support. Thank you very much.
Thank you for nice presentation. I have many questions for you. Not only one question. You said the population of acute exposure reported range from 50 to 96%. Is that data true? It's fine. If you open it,
this is a paper published by Dr. K in 2016. It showed that the rate of occupational exposure cases reported in hospitals in Tanzania is range between 50 and 96%.
Percent of society or population or farmers? Which portion of?
This study was conducted in the district in northern part of Tanzania.
But this is an incredible percent.
Yeah, you see hospital reported cases and community reported cases.
They must have also the people which died from exposure of pesticides. They must have this data too. Do you have any data for that?
Sorry?
The data of that data from exposure because of organophosphatides is very poisonous. And the 15 is so low. For example, in Europe, also in my country, it's prohibited. We are not using organophosphatides in any environment or in any for farming because of the toxicity. You said the acute exposure range, the range is so high. Many people must die from that exposure rates. I think we don't have data for that.
Let's go to the table.
Yeah, I think you can discuss the...
Yeah, and...
if you... and also you can suggest something.
Yeah, okay.
Because
okay.
we have a very limited
okay.
time problem.
Okay, thank you very much.
Okay.
No.
The next presentation belongs to Dr. Ablan. I would like to invite her to make her presentation.
Many body parts, including internal organs such as limbs,
heart, brain, and lungs. Due to high level of the regeneration ability, they are preferred as a near animal models in laboratory studies. The brain of salamander consists of compact lamina of periventricular gray matter, and it is characterized by small number of migrated cells. Axolotl brain consists of different regions: the telencephalon, diencephalon, mesencephalon, and rhombencephalon. It is the image of the dissected brain. The biggest area is the telencephalon area, and between two hemispheres, also there is some structure which is called chorionic plexus. The red circular area is chorionic plexus of the axolotl brain. The neuroanatomy of axolotl brain is similar, have some similarities between mammalian brain tissue. And center of the ventricle, they have the chorionic plexus. We can see the dissected microscope, the part of the chorionic plexus between two hemispheres, and also we can see the chorionic plexus in the ventricular region. And around the ventricular region, cells are organized in layers, and these type of organization is similar to the mammalian brain in a mammalian embryonic brain. A distinctive feature of the salamander brain is the residence of radial glial length-like ependymoglial cells. They are lining in the ventricular region. The white area is the ventricular area, and these ependymoglial cells, ependymoradial glial-like cells, arrange this layer, and the specificity of these animals, these cells, resident in adulthood. The cells have neurogenic potential and have important role in injury-induced neurogenesis in adult brain. They are characterized by long cytoplasmic extension. It is the example of the immunohistochemistry image. These cells labeled with GFAP to analyze the ependymoglial cells. And we can see the one type of ependymoglial cell with long cytoplasmic extension. Radial glial cells are immature cells. In mammalians, the number of radial glial cells gradually decrease as development process and transform into astrocytes. Therefore, it is almost absent in adult mammals. In mammals, radial glial cells transform into the slated ependymal cells, parenchymal glial cells, and adult neuronal stem cells. They produce different types of cells. Ependymoglial cells in salamander, they express markers. GFAP, it is also astrocyte markers, and glutamine synthase and aquaporin. These markers are also astrocyte markers. In my thesis, the second step is metamorphosis. Metamorphosis is the transformation of the spaces into morphologically, physiologically, and anatomically different structures within the same spaces. Thyroid hormones are an important factor in the initiation and completion of the metamorphosis. Frogs also naturally accumulate thyroid hormone, and they, in this way, organs are rearranged and adapt to the terrestrial living condition. Secretion of the T3 and T4 hormones
these are the and the duration of the action varies depending on the thyroid receptor's sensitivity of the tissue. While pre-metamorphosis, neotonic, this line is neotonic axonal brain tissue image, and the line is the metamorphic form. We can see in detail the achromatic and the heterochromatic structure is changed. The neotonic form, their cells looks like the achromatic form, and the metamorphic axonal brain, their neurons looks like the heterochromatic structure. And also there is vacuole in their cell, and their cell shape is changed when we induce the metamorphosis, and the cell of the brain tissue is changed, the neurons is changed. Also, the neotonic and metamorphic axons, they have big nuclei and their cytoplasm very small, very thin. We can see these are the neotonic form of the neurons. They are achromatic cells, and also in the telencephalon occurs in 3 main steps: neurogenesis, migration, and synaptogenesis. Heterochromatin supports the cell migration process by affecting both the mechanical properties of the nucleus and the genetic process occurring within. Condensed chromatin and stiffer nucleus can increase the nuclear resilience to migration stress and significantly reduce the DNA damage during the migration process. It has been shown that there is a significant increase in the heterochromatin levels in the cell nucleus as a result of the stimulation of the cell migration in cell types. It can be said that after metamorphosis, glial cells, they are arranged near the ventricular wall. Their apical surface, they have the big sac, and we can see the achromatin cell nuclei, and the ventricle, these are the light area is the ventricular area. They have the big sac. These cells have also they may have cilia. We know that when a cell produce some products, they generally do not have any cilia, but maybe these cells have the cilia, this small projection maybe belongs to this cell. I have 2 different electron microscopic tissue processing. The first one is the classic electron microscopic tissue processing, and the second one is envelope staining. This type of tissue processing, we use the envelope staining. We obtain these sacs, looks like filled with the liquid, and all of the cells same, but the processing is different. The first image shows us maybe we do not fix the structure. Generally, osmic tetroxides and glutaraldehyde do not fix the carbohydrate. For this reason, I prefer to stain these granules with carbohydrate staining. The first one is a periodic acid shift staining, when we can see these circular shaped products stained with the ASEAN. And also neotonic form, neotonic ependymoglial cells, and metamorphic, all of them stained with blue. Mucopolysaccharides, also called glycosaminoglycans, found in the animal cell membrane and extracellular matrix. They are important molecules in organogenesis, growth control, cell adhesion, signal transfer. Label cells are as shown. These are the neurons. Some of the epithelial, some of the ependymal cells have long cilia, and maybe this area consists of the aminoglycans, glycopolysaccharide. And also, some of the ependymal cells have big cilia, and some of them do not have any cilia. They only consist of the apical surface of the cell. These are ependymal, and also there are many cilia, there are many microvillus around this shape. Our histological and ultrastructural examination show that light and electron microscopic features of these cells, which have a stack at the apical surface, are similar to the goblet cells. These goblet cells, like located in the third ventricle, well, in the axonal brain, may be producing aminoglycans for cell proliferation, differentiation, regeneration without any scar formation, or may be contributing to brain ventricle development and may play a role in brain fluid flow. When cell structure is examined, this structural organization suggests that there may be a transport process from basal to apical or from apical to basal between cell. Light and electron microscopic findings of ependymal cells have shown that these cells located in the ventricular wall consist of subgroups with histological different structure. The presence of the glycopolysaccharide containing secretory filaments was detected in the apical part of the group of ependymal cells. It is thought that these cells may be, I didn't mention about this part, and it was observed that apical surface specialization of the ependymal cells located on the ventricular wall in the telencephalon and diencephalon region of Society for Stereology. Thank you very much.
Dr. Aslan, thanks so much for your very nice presentation. Do you have any questions or?
Then contributing to the viscosity of the fluid in the ventricle.
Skin surface is a mucotic structure, looks like that. Maybe for this reason their brain fluid needs this type of the structure. When we touch with these metal, it changes their form and the solid structure. And it is also, I try to do electron microscopic tissue processing, and it is not fixed with the process. And I cannot see anything. I cannot see the fixed tissue. And when we try to fix it,
around the world. So, and as a result of getting lots of motor functions, lots of sensory functions, another antioxidant is sesame oil. Then we do cross-linking the segment. Finally, you can see the area of touch of the tip. Then we check back the skin and close, then we start treatment with gingival assistant. So, paper, then the print of the ink on the rat setting here after touch. And also we have a microscopic evaluation. So we use also a MAIST counting frame, as mentioned before. And you can see here, this is our MAIST counting frame, inclusion line and exclusion line. Then, if you see here, we are thick with green. This technique, they actually so pick of seasoned oil or ginger oil or the combination between the two substances. Then, regarding the initial thickness, it doesn't reflect any positive effect of substances, but the intention of the tendency, it reflects the positive effect or strong positive group due to crushing, then the effect of ginger oil and sesame oil on the layers after treatment. Promotion of their regeneration. In discussion, we can say ginger oil has very strong protective effect on the peripheral nerve regeneration, oil and sesame oil on the peripheral nerve regeneration. But latency, based on latency, utilization of both is recommended, giving us this opportunity to come to Tanzania and to have a presentation at this conference. Finally, I would like to thank the organization committee of this conference.
They are sensitive to orientation in space. And how did you account for that? And there's basically two ways. Either you randomize the
this tissue was received. This place, if I incorrect, yet we are happy to be here. Thank you very much.
Wi-Fi device.
Distance 33
I think if we use 30 centimeter, 20, 10, 5, then we can examine the effect of distance or which is closer to the Wi-Fi device on the dead guys. Thank you. Thank you.
And the next speaker is the Ömür Deniz from Turkey. Her topic is the from particle fractionator method. Three dimensional counter boxes located in thick sections. This is
get the nerves, proximal and distal to the side of nerve injury to reach the nerve impulses, electrical stimulation 3 volt injury. So the rats were given orally 25 milligram per kilogram daily of dexamethasone for 4 weeks orally. The rest of the short-term group, 24 animals, we made the injuries by the crushing the nerve.
Therefore, the organizing committee also to be inside. Organizing committee.
Şeyler de, Mustafa Beyler de var. 14. Şeye göre normalde 17 kişiyiz gururla, 16 kişiyiz, gurursuz 19 kişiyiz, Kaptanla ve Muhammed İsa'yla. Kaç oldu hocam? Flaşına tıklıyorsun, sana alalım. Sonra bir bilgisayar açayım, sana vereceğim. Tamam.
Okay. Hope now things are in order. I see people almost want to go to Ngorongoro. Great. Thank you very much, Vice Chancellor, Professor Mauricio, and also the co-chairs of the Congress, Professor Levy, and also Professor Kaplan. We want to wind up the sessions, but we are able to do. Not a spokesperson for that. You are the one, but
so thank you very much, and the work of the jury was actually
The second, who also included excellent morphological work, was Dr. Josephine Mapunda. Congratulations, Josephine. And the best sort of presentation for work that was purely done with stereology is presented to Dr. Pankaj Kumar Rathi, who worked on epigenesis of the human fetal kidneys. Thank you very much and congratulations. Please keep up the good work, all three of you and all the others who presented. And please remember, stereology is going to be an excellent way of adding value to your research work. Thank you.
Thank you very much for those who are winners. Congratulations. Now we go for sessions for gifts, zawadi, various gifts, and for everybody, even the photographer. Even photographer. Okay, School of Lisbon. I'm told there's a special gift for School of Lisbon. And Vice Chancellor wants also to say thank you. And Bioengineering, they want again to offer gifts for you too who have come. Now we start again. Professor Kaplan first. This is special from the Dean and the Chair of this. Or we begin with males. The ladies will follow. They have their different gifts. Males, please.
Males, please.
Males. Men. All men. All males. Special of ladies. The two men, please don't go away. Just please stand by. This is just to remember that you were here.
My wife. My wife.
This is okay, please those two men who didn't get the tie, please come. The two men, gentlemen, please come. There were two. Yes? Please. Not yet. And there were two who missed the first round. Yes, there is a very special gift for you today. Is it him? Hussein?
Yes, he is here.
Is it him? Hussein?
Yes, he is here.
Hussein, please send him. And there is Abdurrahman Hassan.
Yes, I'm here.
Okay. Just wait here. I just want to make sure you are here. Murad? Professor Dr. Murad? Okay. Gamze? What's it Hussein? Dr. Abdulrahman, you follow? Yes, you. Professor Dr. Abdulrahman.
Dur seni bir çekeceğim. Şeyle birlikte.
And then Alkan. And then Alkan. I hope this was the last one. Congress.
Mimi nitarudishwa hapa kwa akaniambia
Organizing
kila mtu
committee.
anaona hapa na watu sita. Nikamwambia msimu wetu utaenda zaidi hapa. Zunguka mtu mwenyewe unaweza kukaribishwa hapa. Tofauti wewe amerudi siku amerudi. Sisi tutapata kitu. Hata kwa laki atapelekewa. Yaani akija sasa akija tarehe kusema ndo baba kabisa ameona anambia laki. Sijui laki katika nje. TV tatu laki tano.
Asha Mafita, Sandra Kingazi is not here, I think. Dr. Esther Kimaro, John Semando, Glorieta Magisa, Glorieta is Salina Masawe, session, I want to say...
You haven't announced yourself.
You give. What?
I have to announce you.
My name is Michael Haole. Now, without wasting time, I call Professor Mbega to invite the Vice Chancellor just to official close the Congress. Very good.
Professor Kitanula, Professor Kakan, and distinguished participants, ladies and gentlemen, I'm honored to be here, especially at this very time after interacting with you for 2 days. It has been a very important session for the School of Life Science, and those who attended, we really appreciate the time you all spent. We were many, and this is an area we want to strengthen as we go forward. So the School of Life Science is really looking very much forward to collaborating with you, especially in the area, this area of stereology that we want to strengthen, so solve problems in that particular area. You already know our motto here, the Nelson Mandela's motto is academia for society and industry. So in that very good. Try to connect with you. We probably will a possibility of having a group maybe WhatsApp group, and we can keep our discussion going
if that will be possible, Prof. Kaplan will give an advice, and then we keep our move going and try to interact as much as possible in the future. So without saying much, I just say thank you, but also invite my Vice Chancellor to officially say a few words and then close our session. Thank you very much. Prof.
Professor Kaplan, on all distinguished participants of the 4th Asian African Congress. We were privileged to host. I salute you all. I don't want to keep you long here. I understand that you have had 2 busy days, but quite productive. And I commend. It was like a dream. But then a dream has become true. Professor Abdurrahman, thank you so much. I know it's your 4th time to be in Tanzania. We need to start calling you a Tanzanian. And all other participants, I really thank you for your active participation. The only thing that I want to say this evening is to keep this fire going. Let's keep collaborating. Let us work together. Let us write grants together. If we don't have an MOU with the institution, let us forge one. There is an African proverb that says, if you want to move, let us move as a group. Let us move as a Stereology society. I have no doubt about that. Lastly, let me thank Professor Vega and your team, Sister Mary, and all members of the organizing group. Thank you so much. You have done wonders. I know you had to sacrifice at some point to work extra hours. As we agreed, I'm not going to pay you for working extra hours. But I really appreciate your commitment. Thank you so much. Having said that, let me wish you all safe travel back home. For those who have arranged for safaris, I wish you all the best. And let us keep in touch.
Whom am I should say no?
Good evening. It's a really great time. 2 days we spent a lot of time to discuss very different research subject and new techniques. I hope you are happy to be here. You know, such a scientific meeting, like preparing some meal and then serving your guest. And at this moment, really, this organization committee, this institute organized a well-organized meal scientific meeting, and then they give a lot of feed meal to all brain, and then all learn a lot of the information and some of the needed also is here. And also, I always say, please, if you get some information and the experiences, share with your around.